Hello friends, in this module we will talk about the enzyme topoisomerases. Did you know that topoisomerases are so important for replication that if they are absent, it would lead to incomplete replication and hence cell death? We will talk about the role of topoisomerases in replication and also about the types of topoisomerases. We have seen that DNA helicase separates the two strands of DNA so that each strand acts as a single-stranded template. When the two strands of DNA separate, they must rotate around each other. Helicase could handle this task alone if the DNA were linear and unnaturally short, but closed circular DNA molecules such as the E. coli chromosome presents a special problem. As the DNA unwinds at the replication fork, a compensating winding up of DNA will occur elsewhere in the circle. This tightening of the helix will create intolerable strain unless it is relieved somehow. The class of enzymes that introduce transient single or double-stranded breaks into DNA and thereby allow it to change its form or topology are called topoisomerases. Let us first understand this phenomena of supercoiled or superhelical DNA. The term supercoiling means literally the coiling of a coil. For example, the coiling of the coiled wire of a wired phone handset as can be seen on the screen. Maybe the students of this generation have not seen such a handset. DNA supercoiling is generally a manifestation of a structural strain. Supercoiling occurs when the molecule relieves the helical stress by twisting around itself. Overtwisting leads to positive supercoiling, while undertwisting leads to negative supercoiling. If DNA is in the form of a circular molecule, or if the ends are rigidly held so that it forms a loop, then overtwisting or undertwisting leads to the supercoiled state. In order to understand topoisomerases, let us first understand the term topoisomers, and to understand topoisomers, let us revise DNA topology. DNA is a double helix where one strand goes around the other strand. The linking number is defined as the number of times one strand goes around the other. Thus, if there is a 2000 base pair DNA molecule and it is in the B form of DNA, the linking number is 200 because there are approximately 10 base pairs per helical turn. If a linear double-stranded DNA is present, the linking number can change because the ends are free to rotate. But in a covalently closed circular DNA molecule, the linking number cannot change and the DNA is said to be topologically constrained. In the figure on your screen, the linking number in figure A is 1 and that of figure B is 6. The most common helical structure found in nature is that of the B form of DNA, in which the double helix is right-handed with about 10 to 10.5 base pairs per helical turn. It is said to be the relaxed state of DNA. If we take a circular DNA molecule with 2000 base pairs, the relaxed DNA will have a linking number of 200. Suppose we cut the DNA and give the strands two right-handed twists and then tie the ends, the linking number would now become 202. The linking difference is given by the formula delta K is equal to linking number minus standard linking number. Here, as per the formula now, we have 202 minus 200, that is plus 2. The number of base pairs per helical turn have now decreased and the DNA is said to be overwound. Such a DNA is said to be positively supercoiled. On the other hand, if we give two left-handed twists to the DNA and then tie the two ends, the linking number becomes 198. The linking difference here is 198 minus 200, that is minus 2. The number of base pairs per helical turn have now increased and the DNA is said to be underwound. Such a DNA is said to be negatively supercoiled. Underwound state occurs when the DNA has fewer helical turns than the normal B form of DNA. Two such DNA molecules with the same number of base pairs but having different linking numbers are called topoisomers. All naturally occurring closed circular double-stranded DNA molecules studied so far exist as supercoils. Closed circular DNAs are those with no single strand breaks or nicks. When a cell makes such a DNA, it causes some unwinding of the double helix and the DNA is then said to be underwound. As long as both strands are intact, no free rotation can occur around the bonds in either strand's backbone, so the DNA cannot relieve the strain of underwinding except by supercoiling. The supercoils introduced by underwinding are called negative supercoils. The edges of the narrow groove move further apart. This kind of supercoiling is found in most of the organisms. When the right-handed double helical conformation of DNA is twisted even tighter, that is, it is overwound, then it is called positive supercoiling. The helix begins to distort and knot. The edges of the narrow groove move closer together. Positive supercoils do exist in extreme thermophiles, which have a reverse DNA gyrase that introduces positive supercoils, thus stabilizing the DNA against the boiling temperatures in which these organisms live. Let us see this animation in which the two rubber wires represent the two strands of double helical circular DNA molecule. You can see the relaxed state of DNA. As we try to separate the two strands, the DNA gets overwound and undergoes positive supercoiling. It becomes now impossible to separate the two strands. If you like the content of this video, please subscribe to this channel for more video lectures on molecular biology and microbiology. Let us see what happens now at the replication fork. Unwinding DNA at the replication fork would form positive rather than negative supercoils if no other way for relaxing the strain existed. That is because replication permanently unwinds one region of the DNA without nicking it, forcing the rest of the DNA to become overwound and therefore positively supercoiled to compensate. This leads to a tension or a torque ahead of the replication fork. Unless this tension or torque is released, the replication forks cannot move ahead. The class of enzymes that alters the supercoiling and removes the stress are called topoisomerases. The topoisomerases act by transiently cutting one or both the strands of DNA, as we will see in the coming slides. Topoisomerases are classified into two types, type 1 and type 2 topoisomerases, depending upon whether they operate by causing single or double-stranded breaks in DNA. Type 1 topoisomerases, for example, the topoisomerase 1 of E. coli introduces temporary single-stranded breaks, whereas type 2 topoisomerases, for example, DNA gyrase of E. coli, breaks and reseals both DNA strands. Why is E. coli topoisomerase 1, which belongs to type 1, incapable of providing the swivel function needed in DNA replication? Because type 1 topoisomerases can relax only negative supercoils, not the positive supercoils that form in replicating DNA ahead of the replication fork. Obviously, the nicks created by these enzymes do not allow free rotation in either direction. But DNA gyrase, which belongs to class 2, pumps negative supercoils into closed circular DNA molecules and therefore reduces positive supercoiling. Hence, it can operate as a swivel. Thus, type 1 topoisomerases changes the linking number by 1 each time it binds to DNA, whereas type 2 changes the linking number by 2 each time it binds to the DNA. Further, type 1 topoisomerases do not require ATP for their activity, whereas type 2 requires ATP for their mechanism of action. Let us now first discuss type 1 topoisomerases. The best characterized type 1 topoisomerase is from E. coli. As far as its structure is concerned, it is a monomeric enzyme with 864 amino acids. It is encoded by top A gene. Let us now discuss the mechanism of action. Consider a DNA double helix with 3 negative supercoils. The topoisomerase 1 binds to a part of DNA double helix with linking number 6. The enzyme brings about a nick by breaking the phosphodiester bond. The energy released because of breaking of this phosphodiester bond is stored in a phosphotyrosine bond formed between the 5' phosphate and tyrosine residue present in the active site of the enzyme. The 3' OH end is held in place by binding non-covalently to a domain of the enzyme. Thus, a DNA enzyme intermediate is formed. The enzyme passes the intact single-stranded loop through the resulting gap and reseals the gap. This results in twisting DNA double helical structure by one turn. This time, the phosphotyrosine bond is broken and the energy is utilized to form the phosphodiester bond again. The enzyme dissociates, the linking number is reduced by 1 and the negative supercoils are decreased by 1. Let us quickly revise this same mechanism of action of type 1 topoisomerases. The enzyme binds to the DNA duplex and brings about a single-stranded cleavage. It then causes the intact strand to undergo passage through the nick. The nick then undergoes ligation and finally the DNA is released and the enzyme is free to bind again. Let us now discuss the other type of topoisomerase, that is type 2 topoisomerases. The best characterized is E. coli topoisomerase 2, known as DNA gyrase. As far as the structure is concerned, DNA gyrase is a tetrameric protein with 2 A subunits and 2 B subunits. Depending on DNA substrates, they can change positive supercoils to negative or increase the number of negative supercoils by 2. Topoisomerases 2 also catalyze catenation and decatenation, as we'll be seeing in the next slides. The enzyme also introduces negative or positive supercoils at or near the orifice, that is the site of origin of replication. It also removes positive supercoils ahead of the replication fork. Let us now discuss the mechanism of action. Hydrolysis of ATP by ATPase activity of gyrase causes a conformational change in the enzyme. Initially, enzyme binds to one part of the DNA, the G segment of DNA, inducing conformational changes in all the A and B subunits, as can be seen in step 2. After binding the ATP and another strand, T segment, a series of reaction occurs in which the G segment is cut by A and A prime domains of the enzyme and the ends of the G DNA become covalently linked to the tyrosine residues in these domains. The binding of ATP and T segments induces a series of conformational changes. The G segment is cleaved and the resulting two fragments are spread apart by at least 20 angstroms. Simultaneously, the ATP binding domains move towards each other and they dimerize, transporting the T segment through the break, as you can see in step 4 on your screen. The DNA transport step is shown as proceeding through the hypothetical intermediate step. The cut G segment is resealed and the T segment is released by a conformational change through the lower gate of the enzyme subunits. This gate then closes as the ATP is hydrolyzed and the resulting ADP and PI are released to yield the enzyme in its starting state, that is the second state, as which can second step, which can be seen on your screen. Alternatively, the enzyme can now proceed for another cycle. Thus, topoisomerases have a very important role to play in relaxing the positive supercoils formed ahead of the replication forks. Topoisomerases have another role assigned at the end or the termination of replication. DNA replication often results in two completely or incompletely formed daughter duplexes intertwined or interlinked with each other. These interlinked circular DNA molecules are called catenanes, and separation of these catenanes is called decatenation, and this is brought about by the action of topoisomerases. Let us delve a little bit more deeper into this role of topoisomerases of decatenation. If the end result of termination of replication is two incompletely formed daughter duplexes interlinked to each other, then they can undergo decatenation first. For this, a single-stranded nick is required, and this can be brought about by type 1 topoisomerases. The two incompletely formed daughter duplexes get separated. This is then followed by a repair synthesis so as to yield two completely formed daughter duplexes. Alternatively, two incompletely formed daughter duplexes can undergo repair synthesis first so as to yield two completely formed daughter duplexes interlinked to each other. This would be followed by decatenation, where a double-stranded nick would be required, and this could be brought about by type 2 topoisomerases. With this, we come to an end of this lecture. In this lecture, we have talked about DNA topoisomerases. We have first revised DNA topology, and then we have talked about DNA topoisomerases 1 and topoisomerases type 2, and we have seen how they are responsible for relieving the tension and the torque which is present ahead of the replication fork, which is caused as a result of the supercoiling of DNA. We have also talked about the role of topoisomerases in decatenation, that is, which occurs at the termination of replication. We'll be meeting again in the next lecture where we'll be talking about the replication process. We will start with initiation and then we will move ahead with elongation and termination in the coming lectures. So stay tuned to this channel and subscribe to this channel. Thank you.